So we know that we have something called an ABO blood group. So what this ABO blood group is representing is the different types of surface antigens. Okay, so we'll use the term antigen, all right, which is a surface marker on the surface of different cells. So we can have self antigens that are part of our own body that are normal to us and recognized by our immune system as self. And then we have foreign antigens. So in this ABO blood group that we're going to talk about, we're talking specifically about self antigens, right? So in the context of blood, these antigens are also called agglutinogens. Agglutinogens, all right? So this is the same thing as antigens when talking about our blood, right? Now, the opposite, or the not necessarily the opposite, but the complementary thing that goes along with antigen is antibody, right? So we can have antibodies, all right? The other term for antibody when talking about our blood groups are agglutinins. Agglutinins. So we have agglutinogens, which are the antigens, and we have agglutinins, which are the antibodies, right? So when looking at our blood cells, we know that on the surface of our erythrocytes, we're going to have different, let me draw a better circle here. I'm going to draw it actually down here. We're going to have different blood types, and these blood types are dependent upon the presence or absence of certain surface antigens or surface agglutinogens, all right? So we're going to call these type A blood. We're going to call this one type B blood. This one here, the third one, is type AB. And this fourth one is type O blood, all right? So it turns out that on the surface of some of our cells, we have antigens that designate as either being A, B, or O. So on the type A blood, we have A antigens. So I'm going to denote these in blue, right? So on type A blood, we have A antigens. And in type AB, we also have A antigens because we have the A in AB, all right? We also have B antigens. So in type B, we're going to draw B here. And in AB, we also have a B right here. So in type O blood, it turns out that O, there's not an O antigen. O just means we don't have any. Okay, there's not an antigen present. So you don't draw and you don't represent any antigen on the surface of the cell. Okay. Now there's another thing that goes along with our blood type, and you likely have heard this, is the RH factor. And this RH factor is what denotes our blood as either being positive or negative, right? So we can have A, positive or negative, B, positive or negative, AB, positive or negative, and O, positive or negative, okay? So this RH factor is sometimes also called the D antigen, okay? Because it is encoded by a gene called the RH gene or this RHD gene, all right? Now, the RH factor was originally discovered in the rhesus monkey. And so the RH is an indication of that, all right? So in A, if we were to have type A positive, we would have the D antigen on the surface, okay? Or we'd have that RH factor, all right? If we are negative, then we don't have it, right? So negative just means that it is not present, right? Now, these are the surface antigens, right? So the type of blood, A blood, B blood, AB, that is indicating what antigens or agglutinogens are present on the surface of that red blood cell, right? Now, going along with the antigens, we also have corresponding antibodies, all right? So just like we said here, we have agglutinins. So it turns out that we have an opposite antibody for whatever antigen that we have. So if we have type A blood, we have anti-B antibodies. All right. So this means that if type B blood is introduced into somebody with A blood, their anti-B antibodies are going to attack and form a complex with the B antigens of that B blood. All right. So if type A blood has anti-B antibodies against type B, what does type B have? Type B has anti-A antibodies, right? Because it is opposite. So if type A blood is introduced into somebody with type B blood, their anti-A antibodies will attack those A antigens, right? So if we have type AB blood, what type of antibodies do you think we'll have? Well, it turns out that we'll actually have none, right? We'll have none because we have both antigens, right? We have the A antigen and we have the B antigen. So there's no antibodies left over. If A blood is introduced, it will accept it. If it is B blood that is introduced, it will also accept it, right? So using that information, if we go to type O blood, what antibodies do we have? Well, because we don't have either antigen, A nor B, we have both anti-A and anti-B antibodies, right? So this means that if A blood is introduced, it will attack it, the anti-A antibodies of type O blood. If B blood is introduced, the anti-B antibodies will attack that B blood, all right? These antibodies, this is an important to note about these antibodies, they are naturally occurring in the blood, in the plasma of their respective blood types. Always, you don't have to be exposed in order to develop them, okay? So if we're looking at type A blood, just as an example, right? So let's look here at type A blood. Say type B blood is introduced. Those anti-B antibodies are already there. They don't have to work to be developed. They don't have to be formulated upon the arrival of that B blood. They're already there. In the event that B blood is introduced, they can then work against it, all right? Now, that is not the case for the RH antigen, all right? So let's say we look at type O blood over here, all right? So let's take a look here at type O blood. Say this is O negative, all right? So we're looking specifically at O negative. Because it lacks the RH antigen or that D antigen, that does not mean that it naturally has the anti-RH antibodies, right? So there are what are called anti-D or anti-RH antibodies, okay? But these are not always there. They're not innately there from birth, okay? So if they're not innately there, then how do they get there? How do we develop these anti-RH or anti-D antibodies? Well, we have to be introduced to blood that is Rh positive in a term called sensitization, right? Sensitization. All right. So what this means is that you have to give Rh positive blood to somebody that is Rh negative. When that occurs, the person will develop these anti-Rh antibodies, and then usually upon the second exposure to Rh positive blood, they will formulate a response against that blood. We'll have a separate video about something called hemolytic disease of the newborn, in which this case happens, in which this event happens, where Rh positive blood from the fetus mixes with the Rh negative blood of the mother. And during the first pregnancy, it's usually not an issue, but in subsequent pregnancies with an Rh positive fetus, it can actually lead to issues. All right. So now that you know the basis of these blood types, we can begin to look at and assess examples of testing blood for certain types. So if we look down here, we have a nice little chart here. All right. So this chart is showing a kind of control blood sample here. All right. And then we have a blood type or a section of blood here that was given anti-A, anti-B, and anti-D serums. All right. So what this means is that there is a drop. So say this was person A, this is person B, this is person C, and this is person D. Okay, so these are just kind of arbitrary designations. This one up here is person A, person B, person C, person D. All right. So one drop of person A's blood went here, and then another drop of their blood went here, and another drop of their blood went here. All right. So let's take a look first at, let me erase all of this, clean this up a little bit. If we look just at this section right here, all right, what we add is we add something called anti-A serum, okay? This anti-A serum is not the same thing as the anti-A antibodies, all right? What this is saying is if we add this anti-A serum to this drop of blood and it reacts and you get these little these things form here in this process called agglutination, then that means that person has the A antigen in their blood, okay? If you add the anti-B serum to this column and it agglutinates, that means they have the B antigen. And if you add an anti-D serum to this column of blood, then that means that person has the D antigen or the RH antigen, okay? So knowing that, you can then identify the blood types of each of these. So if we look here, let me erase this here. So if we look at this column here first, so we look at this column and we're going to look at this set of blood. All right. So in this first anti-A, we see that it agglutinates. Okay. So that means this blood is at least A. It has something with an A. It's either A or AB. All right. If we look at the second one here, we see no agglutination. Right? We see no agglutination. So that means that when the anti-B serum was added, it did not react with the blood because there were no B antigens. So that means this person's blood does not have any B. So that means it's not B blood and it's not AB blood. So we know that we're dealing with type A. And then when we look at our final blood drop here, we see that when we added the anti-D serum, it did agglutinate. So that means that the D or the RH antigen is present. So that means that this is A positive blood. All right. So let's do the second one. And you feel free to pause the video and try to figure these other ones out on your own. All right. And I'm going to continue. So in the first column, we see no agglutination. So we don't have A. In the second column, we see agglutination. So we have B. And in the D column, we see that it agglutinates. So this is going to be B positive. Okay. We look at this third row. Here. In the first column, we see that it agglutinated, so we have A. Second column, we also see that there's agglutination, so that is B. And in the third column, we see that there's agglutination, so this is AB positive blood. And in this fourth row here, we see that we do not have agglutination in any of the wells, any of the blood, so we know this is O negative blood. All right. So with this, we get and of these examples here, two of them are very significant. All right. Particularly, we're going to look at AB positive and we're going to look at O negative. All right. So if we come down here and we draw a blood cell, let's draw a red blood cell, and we make it AB positive. All right. So here is an A antigen. Here is a let's do it in green. Here's a B antigen, and then in black we have a D antigen, right? So this blood type is AB positive, all right? So if we have A antigens, B antigens, and D antigens, what antibodies do we have? Okay, so let's ask ourselves, what antibodies do we have associated with this blood type? Well, you have the opposite antigens of whatever opposite antibodies, whatever antigen you have. Well, we have all the antigens, so we have no antibodies. Okay, so if there are no antibodies, what does that mean in terms of what blood this person could receive? Well, they could receive all blood types, right? Because if you give A blood, there's no anti-A antibodies. If you give B blood, there's no anti-B antibodies, and there's no anti-D or anti-Rh antibodies because they're Rh positive. They already have it. Okay. So what that means for this blood type, this AB, that means this blood type is what's called the universal recipient. Okay. Universal recipient, meaning this blood type is the one that can receive all other blood types. Okay. It can receive all other blood types because it contains no antibodies in its plasma in the blood that would form a reaction with any other blood's antigens. So this is the universal recipient. All right. So if this is the universal recipient, let's now look at the other one. So this is going to be O negative blood. All right. So O negative, remember we have the opposite antibodies. Okay. So in this case, we're going to have what? We're going to have anti-A antibodies, and we're going to have anti-B antibodies. Okay. Because, and remember, we're not assuming that we have naturally present anti-Rh antibodies. We don't know if this person has been exposed yet. So we're just going to say that there's none there. So if we have no antigens, right? So no A, B, or D antigens on the surface of this red blood cell. That means this red blood cell and this blood type can be given to anyone else. Because no matter who you give it to, there's no antigens there for the host blood to attack, right? You could give this to A blood and the anti-B antibodies aren't going to attack because there's no B antigens. You could give it to the B blood and the anti-A antigens wouldn't attack because there's no A antigens, right? So this is what is called the universal donor, right? Universal donor, meaning this blood can be donated. It can go to anyone else because of the lack of antigens on the surface of this red blood cell. Okay, so that wraps up and concludes our blood lecture. If you have any questions, please reach out. Again, think about the surface of the red blood cell. Do we have AB, or nothing? What does that mean for the blood type? Look at how to identify blood types right here and gain some practice. And then understand how the different types of blood, the AB, the O negative, can be labeled as a universal recipient, universal donor, and what is the reason for that. All right, thank you.